Yes, we can. So, welcome everybody to this very general session that is called I think as you work on soil, you all know how important soils are. Myself, I'm Ludger Hermann from Hohenheim University. I'm a pedologist. I'm working mainly in tropical agriculture area, and that's probably why I was asked to host this session. We have 5 presenters, from which I see 3 on the screen. I shortly introduced you to the rules. Every presenter has 10 minutes, so that there are about 5 minutes left for discussion. And at the final end of the session, again, there will be some time for a generalized discussion on the different topics. So now I do not want to make any further words. I think the content is more important than having me talking here on the screen. So I would like to invite Mr. Zhu to speak about the effects of climate on decomposition of cattle, sheep, and goat manure in Kenyan tropical agriculture. So welcome to Eastern Africa. Mr. Zhu, you have the panel.
Okay. Thank you for the introduction. I'm Yuhao Zhu from KIT IFO. I'm going to talk my part of my PhD study, the effect of climate on decomposition of cattle, sheep, and goat manure in Kenya tropical pasture. This work is supervised by Professor Klaus Butterhabao and mainly conducted in livestock, International Livestock Research Institution in Kenya. So first, let's talk about why we focus on the decomposition. The decomposition is the physical and chemical breakdown of dead organic matters, and the plant later decomposition is very important for carbon and nitrogen cycling. For example, more than half of the net primary production is returned to the soil through the later decomposition. It also results in CO2 emission to the atmosphere of about 6 pg carbon per year. And for the nitrogen cycle, internal recycling of nitrogen from later decomposition is also the primary source of nitrogen for most ecosystems. However, in the grazing system, like grazing animals consume large amount of biomass that often reduce the later input to the soil. Instead, the consumed biomass cannot be digested by animal totally. So large amount of the biomass will be returned to the soil in the form of the manure. So manure is important for carbon cycle in grazing system, but not so many studies is available. This is especially true for sub-Saharan Africa due to the farm type, smallholder mixed group, and partial livestock system. This farm type is normally small, with only 0.5 to 2 hectares, and mainly rely on pasture feed, long-time daily grazing, and seldom have any nitrogen fertilization. So in this system, the manure are very important for to maintain and keep the productivity of the grassland. So how about the manure decomposition? We have to better use the manure to keep and maintain our grassland. From previous later study, we are already know the plant later decomposition follows the exponential decay patterns and mainly influenced by the later quality, climate, and the decomposed community. How about the manure decomposition? The completion will be the same like later? So following this question, we are aiming to measure the changes in manure dry matter, carbon and nitrogen concentrations over time after manure decomposition, and determine if manure type like manure from different animal species affect manure decomposition rates, and determine how climate affects manure decomposition. Uh, Based on our objective, we hypothesize manure decomposition will also follow exponential decay. Manure decomposition rate would be faster for manure with lower initial carbon and nitrogen ratio and higher initial nitrogen concentration. Manure would decompose faster under wetter and warmer climatic conditions. To achieve our objectives, 3 different manure types are selected. Cattle, sheep are good. Rhizosphere animals are widely grazed in sub-Saharan Africa. Applied to 4 different climatic condition sites: Rim, Theta, Kapiti, and Elri. These sites the precipitation are increased from Embu to Elri, but the temperature, the annual mean temperature are decreasing. Also, normally the cattle manure has higher C ratio and lower nitrogen concentration compared with the sheep and the goat. We use manure bags. We collect and weight the fresh manure and put it into the litter bags. Then after weaning, we put the bags into different sizes. Then simply weekly, bi-weekly, or monthly over more than 1 year. This is the result. After we collected the samples, we find that the remaining dry mass following the exponential decrease and also the carbon content in the rest dry mass. However, the nitrogen concentration kept constant or a slight increase, which means the nitrogen mineralization is far slow compared to carbon mineralization, which also related to the decreasing C ratio in the manure bags. Then we also calculate the dry matter decomposition rates, considering the original dry mat and the rest dry mat, also the time. From the decomposition rates result, we can compare the decomposition rates among different manure types across different sites, and we find if we focus on one manure type across different sites, we find all the manure at every site always decompose faster. And if we focus on one soil, at one site, but a different manure type, we find always the cattle manure is decreased faster than goat and sheep. So this means both manure type and climate will influence manure decomposition rate. But contrary with our hypothesis, the cattle manure had a higher C ratio and lower nitrogen concentration, decompose faster. To explain this, we also measured the manure chemical characteristics, including crude protein, lignin, cellulose, hemicellulose, and we find the cattle manure had a higher cellular concentration, and during this process, the cellulose decreased the most. So cattle manure has the highest cellulose and the cellulose decompose fastest, which led to the cattle manure had the highest decomposition rate. And we also build a relationship between the manure cellulose and nitrogen ratio, and find the cellulose and nitrogen ratio show the positive relationship with the decomposition rates. For the effect of climate factors, we find among different sites, the cumulative precipitation show the positive relationship with the decomposition rate. To better synthesize the native environmental condition, we also calculate the aridity index based on the equation precipitation divided the potential evapotranspiration, and also built a positive relationship between the aridity index and the decomposition rate. So Rainfall amount, limited manure decomposition, and tropical climate. From this study, we find we can conclude that the manure decomposition was mainly influenced by the property and the climate concentration, especially the cellulose concentration and also the rainfall amount. This work was mainly conducted at Maringala in International Livestock Research Institute in Kenya. Thank you. Okay.
Wow, that was perfect in time. Thanks a lot for this very clear presentation. And actually, the floor is open to any kind of questions from the crowd. Any listeners who have a question with respect to this presentation, then please note it down in the question and answers section of your client.
Are we allowed to ask, or just type as a presenter?
As a presenter, you can also ask directly.
Yeah, okay. Yeah, I just have a very small question about the N mineralization. You mentioned that the carbon mineralization was faster than N mineralization. Any explanation? You have any explanation for that?
For this one?
Yeah, somewhere in, yeah.
We didn't measure, we only measured the nitrogen content after like a weekly or 2 weeks. Then we compared, we find the nitrogen concentration keep constant while the carbon concentration decreased. So we conclude that the carbon are more easily lose compared with the nitrogen, and the only way the carbon and the nitrogen can lose is the mineralization. So we conclude that the nitrogen mineralization is much slower compared with the carbon mineralization.
Sorry,
because to my not. Oh, okay.
Sorry, I disagree. So
Yeah,
I think the answer is simply already in this general sketch that you have showed us. Perhaps you can repeat that one. Because what we then see is that the microbial biomass is growing fast at the early stages.
Yeah.
The microbial biomass has a CN ratio of about 1:8.
Yeah.
So what actually is happening is that there is N mineralization, and I think it's as fast as the C mineralization. The difference simply is that the N is stored then in the microbial biomass, whereas the C is leaving the system.
Yeah.
Okay.
Yeah, exactly. And then by the process of which the microbe mine for nitrogen to have the required demands for the activities. So the microbe work fast and harder on the carbon to obtain nitrogen. And in most cases, this nitrogen will be above the demands. One other thing about the climatic factor, did you consider to use the mean annual temperature or just the aridity index?
We use the cumulative precipitation. And
not mean annual temperatures, only precipitation.
I didn't show, but in our study, we have four study sites, and the temperature range is from 80.9 to 22. So, but the precipitation varied from 200 to 1070. So the precipitation is the main difference. And consider the first question, I have 1 point I want to mention about the microbiome. In the manure, the carbon and nitrogen ratio is quite high. Like for cattle is around 20 or 25 in sub-Saharan Africa. So it's not so easily used by the microbiome, because the best is around 5 to 10. If the
Sure, it's the best. Nevertheless, you would not have a decrease in biomass if there wouldn't be decomposition, and the decomposition is mainly finally by the microbes.
Yeah.
So there's no way out.
Mm-hmm.
Okay, just a question for clarification with respect to the methods. So you used litter bags, and you left the litter bags on top of the soil. Is that correct?
Yeah.
Okay. And what was the mesh size? So did you exclude actually mesofauna or not?
It's the mesh size is 1 micrometer. So it's only available for the microbiome, but to prevent the soil animals entering.
You said 1 millimeter, you mean, not 1 micrometer. There's no mesh size of 1 micrometer. 1 millimeter.
Yeah, 1 milliliter.
1
millimeter. Mm-hmm.
1 millimeter mesh size.
Yeah.
Yeah, but that means for certain mesobiota, actually, there would be a chance also to get access. That's just what I wanted to know.
Okay.
Thank you. Any other questions from
audience?
Any have been asked from the presenters? Any further question?
Yeah. Just a quick one. So the litter bags, you said you place them on top of the soil. I was just wondering if, can you hear me?
Yeah.
Okay. So did you place the litter bags on top of the soil? That's what you said, right?
Yeah, we cut the grass and put on the top of the soil, then leave for 1 year.
For 1 year.
Yeah.
Do you think if maybe you had buried them at a certain depth, would that have changed the dynamics? And what do you think would be possibly be the difference?
Pardon, I didn't get.
Assuming you had buried them at a certain depth, because most often when farmers apply this manure, sometimes it's followed by plowing. So the manure is not really directly on top of the soil. Sometimes it can be buried at maybe some few centimeters deep the soil. What would have been the difference?
Yeah, because in my study, we simulate the down and the down patches from the grazing system. We want to know mainly from the grazing system, so it's grassland, not from the level land, from the agricultural land.
Okay.
So I also saw some study. If you really put the manure bag several centimeters deep, the decomposition rate might be high.
We have to stop the discussion here. If there's anybody who has questions later on, we can post. Thank you very much, Mr. Zhu, for the clear presentation, and I hope the next one will also be in time. So I would like to invite Dr. Abdallah, who is working in collaboration with the University of Bayreuth. He is going to talk about patterns and drivers of soil carbon dioxide, well-watered and drought-stressed silage maize. Please, Dr. Abdallah, you have the.
Thank you very much, Mr. Chairman. I will start by.
Don't run.
Huh?
Don't run.
Attend your session.
Okay, hello everyone. As Mr. Chairman mentioned that, my name is Abdullah. I'm working currently as a postdoc at the University of Bayreuth, funded by Alexander von Humboldt's Foundation. And my presentation is the patterns and drivers of soil CO2 emission from well-watered and drought-stressed silage maize. I will move a bit quick here in this slide, like a background of my topic, and I will try to explain the diagram I have on this side of the screen. So everyone knows that the global warming is getting higher and higher, and this is resulting in drought and heat. Is a quite frequent and severe drought and heat, which in turn affect crop productivity or decrease crop productivity, and this results in low carbon allocation to the soil. The carbon allocation to the soil usually take one of the two passes, like the later inputs from the leaf or root, or the root exudation. And then we have down here, we have the microbial activities, which start degrading the litter using the exudation as the substrate. And all this process increasing the soil organic matter decomposition, resulting in high CO2 emission, which is going back to the global warming. So there is a need to look for crop genotypes or land managements that can tolerate the drought effects. And to look at the bottom of this issue, we have to look at the drivers and the mechanism of the carbon dioxide emission to the soil, to the atmosphere, sorry. And this mechanism I have subdivided in like 3 categories, which is soil temperature and moisture content, substrate availability, and microbial biomass and activity. And mainly at the optimum soil temperature and moisture content, then if there is this substrate, that means there is a microbial growth and activity. And this was only influenced or introduced by a mechanism which called rhizosphere priming effects. In short, the rhizosphere priming effect is a change in the rate of soil organic matter decomposition in the presence of the living roots. If this is symbol graph or diagram that explain the rhizosphere priming effect. So if you comparing to unplanted soil to planted soil, or to do planted soil, we have 2 ways of priming effect. Either is a positive way, and in this case, we have more CO2 emission in the atmosphere, or negative way. This way, we have less emissions. And the direction, whether it's a positive or negative, that depends on the soil factor, the substrate themselves, and the optimum growing condition for the crop and for the microorganism. So in this study, we hypothesized that soil temperature and moisture content directly affect microbial activity, whereas drought stress was recently assumed to increase root exudates, accelerating soil organic mineralization, and thus increase CO2 emissions. And the objective of this study is to study soil CO2 pattern and drivers and the well-watered and drought stress /maize. And our experiment is really an extraordinary experiment. It's a Lysometer experiment when we have the greenhouse that move to exclude the rain to make the applicability of implementing drought treatments. And this picture here is showing the Lysometer, drought maize, and watered maize. They are very different. We install like 3 temperature and moisture content sensor along the Lysometer, 1 meter depth at 3 different depths to continuously measure soil temperature and soil moisture. And on the top soil, we are measured using a portable thermometer. And here in this picture, we are showing that we are using the LICO for the carbon dioxide emission, and then the syringe method to take the sample, gas sample for isotopes. And this is the cables for these sensors. So the treatments are mainly well-watered, drought-stressed, and unplanted control with 5 replicates. The system is occupied by drip irrigation to adjust the well-watered and drought stress. And at the end of the season, the soil and the plant were sampled for the lab analysis. So in my results section here, my first figure is showing the differences, which is obvious that we will have high above ground biomass in well watered than the drought stress, and the difference was significant at 27%. And then on the other one is the root distributions along the soil profile, and we can see that the drought stress have more distributed roots, and this could be explained by the maize under drought stress has like a high root system and try to reach the water from any side. My second figure is showing the pattern on CO2 emission. So the red color is showing the water, and then the black is the unplanted control, and green is drought stressed. And we can see during the growing season, there's a lot of variation on the CO2 emission, but it's constantly higher in the water stress. When after harvest, this is a harvesting time, and then after harvest, there is no significant difference on CO2 emission after we remove the maize. And here I'm showing the soil temperature at 10 centimeter depth and soil water content at 10 centimeter depth as well. So the CO2 emission increase with a decrease of soil temperature and increase of moisture content at 10 centimeter depth. In overall, the well-watered maize had 20% and 24% higher CO2 than drought stressed and controlled, respectively. Here on this PCA analysis, I would like to show the drivers or the controlling factors. So the first PCA is unplanted control, and in unplanted control, soil CO2 emission is mostly controlled by microbial biomass and microbial biomass carbon and nitrogen, which alters by the soil temperature and soil moisture, like the top soil temperature and top soil moisture. It can be seen here, is M5 and T5. When I come to the drought stress, so I'm referring to temperature with T and moisture content with M. So here the controlling or the drivers are slightly different, because under drought stress, the CO2 is strongly controlled by above and below ground biomass, and also by microbial biomass carbon and nitrogen. This means that when we irrigate, the crop is stressed, and then when we irrigate little amount of water, the plant start fast recovery. Then the roots and the microbe start to recover fast. That's why the CO2 emission in the CO2 that can emit during this time is referred to the plant biomass and microbial biomass. And the well-watered conditions, we have soil temperature and soil moisture are the main driver for the CO2 emission, and which is clearly. But the interesting thing, we have higher rhizosphere priming effect. The rhizosphere priming, the higher rhizosphere priming effect in this in well-watered treatments, this is defeating the hypothesis. Higher rhizosphere priming effect in this it is defeating the hypothesis that the drought stress increase the rhizosphere priming effect. However, we are not sure yet, because I think there is more thing need to be done to clarify the conclusion, and this is specifically is about the root exudations and also separating the CO2 source. Probably in a well-watered treatments, mostly the CO2 emission coming from the roots, not from the organic matter decomposition. To be clear and to be clear and confident about our answer, we need to include carbon and nitrogen, other microbial activities such as end mining and roots exudation from the plants to confirm this preliminary finding. I can see you appear now, and I think I'm done. Thank you.
Okay, thank you very much. We have been nearly in time, so that's still okay. Thanks a lot for keeping the schedule. And could you please switch off your presentation mode?
Okay.
Any kind of comments?
Yeah, I'm stopping. Okay.
So, anybody in the crowd, please ask your questions via the question and answers function. At the moment being, I don't see any.
Yeah.
So, any question from the presenters?
I think there's. This is chat. Maybe there's something that we don't know whether everyone can ask.
Yeah, but I think that must have been explained before the sessions, that you can use the question answer function of the WOBA app.
Okay.
Yes, under resources you can see session Q&A, and that's where you can pose your questions. So please do not hesitate to pose your questions online. The other presenters. If not, I have a question, and that
okay.
I didn't really get what was of the control. So you have an unplanted control. So that's one factor that is changing. That is not having. But what was the water regime of the unplanted control?
Oh, okay. This is a really good question, because I'm having two controls actually, like a drought control and will water it control. And then with the drought stress treatments, I have to compare it to the drought control, receiving the same amount of water. And with the well watered, I have to compare it with the well watered control, considering the amount of water in both.
Okay, and that are the data that you use for the PCA analysis, yeah?
Yeah.
Okay, so you always just compared with a direct control with respect to the water regime.
Yes, yeah. And this is also a very interesting question. This is also needing for calculating the rhizosphere priming effect, because this is the way we separate the CO2 coming from resulting from the root respiration and the CO2 resulting from soil organic matter decomposition.
Did you consider to measure DOC, so dissolved organic carbon, as one kind of possible analysis that could give an indication for these root exudates?
Yeah, to be honest, yes, we have some other master student actually working on that, because the lysometer are accessible from the cellar, and we are collecting. And to be honest, the experiment about all the carbon and nitrogen budgets, like the input and the outputs, but I think the time is not enough to present all of this. But I think analyzing that in the water, it can give an indicator. But I think the best way is to plant in the controlled condition and to try to extract the exudates using a proper method, quantify.
Actually, I think you in your message sections you mentioned Yasha Kuziakov.
Yeah,
and he tried to identify these kind of shares by labeling with 13C the exudates, for example. So that could be also an idea.
Yeah, I'm also working with Prof. Pausch. He was a student of Jacob. But then labeling in the field is quite a process.
Sure.
Yeah, it's a very hard task to do labeling. We are thinking about it. Hopefully, we can manage. We're just trying to find an easy way to do a field labeling, and then in such a way, then we can check in the leaches because we labeled the plants.
Yeah, that's it. Yeah. Okay, fine. Any other question from the crowd or from the presenters?
Okay, for all of you, the Q&A is not functioning because of technical failure. That is why no question come to you. Okay.
Ah, okay. So I haven't seen that. Thanks for informing me about that one. Okay, so no questions from outside. It's really a pity for the presenters.
Yeah,
and for jobs. So we have more time for the single speeches. Now I have to check again. Actually, the third presentation would be by Mr. Roldan Torres Guterres. And I don't see him here in my list. So it doesn't seem that he is present. If you are present, dear Mr. Roldan Torres Guterres, please raise your hand. If not, I have to assume that you are absent and that you are not going to present. Okay. So then, if that is the case, then...
Yeah, I think I'm just receiving some question from the app. So I think every presenter may be asked to check his app. Okay. Okay, so you're waiting.
Probably we can postpone his presentation. Then we would have presentation number 4. That is from Mr. or Mrs. Sorry that I can't identify. Asheri Yayu, or no, Natalie. So it should be a woman. Natalie Asheri Yayu. Are you present? So no sign from her. Then we just go on to the 5th presenter, and I would like to ask Sebastian Ayin Bisa Yanore from Ghana, I think, whether he is ready to share his presentation. Sebastian, are you ready?
Yes, I'm ready.
Oh, that's marvelous. Thanks a lot for this favor. So you are going to present on gravel content of soil impact on water use, growth, and yield of pearl millet. Actually, pearl millet is one of my favorite crops. So I'm very interested in hearing what you have to contribute. So you have the floor.
Having some technical issues. Can you see my slide?
I think I can see your screen.
Yeah.
But not your slides.
Okay.
No.
Wow.
Is it a PDF?
Just 1 second. I will.
Yeah.
And now?
I still can't see you, can only see you.
You are back to the Zoom meeting, but we don't see your screen. At least.
Yeah.
There's a button normally on the
I see that.
center of the screen down.
Yes, I've seen that.
It's starting. Now it's working.
Yeah, okay. Now we have it.
Okay.
Okay. Is it? Can you see it now?
Yes, definitely.
Okay. So I start. Yes. My name is Sebastian Ayimbe Sayanori. I'm PhD student at the University of Ghana, Department of Soil Science. And this evening, I'll be talking to us about the gravel content of soil, impact on water and fertilizer use, growth and yield of pearl millet, Penicitum glycum. And there are some co-authors of this work. That's Samuel Kosi Goffred Adiku, Dele Sefako Makati, Daniel Ese Dodo, and John Bright Amwa. Population has increased, and this has pushed marginal lands into agricultural production, and the use of fertilizers have also increased. This does not exclude the use of fertilizers on marginal lands. And what are gravel soils? Gravel soils are soils that contain more than 15% by volume. In northern Ghana, 50% of all the soils are classified as gravel, and about 25% of soils in Ghana are also classified as gravels. And these gravels ranges between 25 and 80% by volume. To illustrate the problem with gravel soils, let's consider these sketch as three columns, with the first column being a column without gravel. So this is a gravel-free soil. The second one is a column with small size gravels, and the third one is a column or a soil with larger size gravels. So the challenge with water is that water moving through a soil, a gravel-free soil, the water movement is through the soil matrix only. But when it comes to the soils with gravel, so when the gravels are small in size, then these gravels become an impedance to the movement of water, because the active area available for the movement of the water is reduced. That means that the tortuosity of the water flow also increases. And at the long run, it takes a longer time for the water to move from point A to point B. Whereas in the case of larger size gravels in the soil, the movement of water is very fast because of the creation of macropores due to the presence of these gravels. In case of fertilizer, a similar trend happens. So soils without gravels, the main driver of fertilizer movement is through the soil matrix. And whereas when soils have gravels, the gravels create macropores, and these macropores make the movement of fertilizer and solute very fast, and then it's short-lived in the soil. So these solutes move beyond the root zone of plants, and they are easily leached to levels where plants cannot make use of. So these are challenges that we set up to investigate. And the objective of this research was to quantitatively describe the effect of soil gravels on water and fertilizer balance components, the growth and the yield of pearl millet, but to also look at the water and fertilizer use efficiency in these gravelly soils. To do this, we set up a greenhouse experiment using a 4-factorial design. We sampled 2 soils from northern Ghana, Utric Leptisol and Ferric Lexisol, using undisturbed monoleaves of PVC pipes of height 45 centimeters and diameter 16 centimeters. 18 columns of the undisturbed soil samples were taken. And then we also took soil samples. Those are disturbed soil samples. We sieved them and we repacked them using similar columns with varying gravel content. And we had 24 columns of the disturbed soil samples. The table here represents a summary of the different fragments of gravels that were contained in the different columns that we used for the experiment, with gravel of 20 to 30 millimeters having the largest fractions or having the largest percentage in each column. These columns were assembled in the screen house and planted to millet and fertilized at 100 kilogram per hectare and 40 kilogram per hectare P and 40 kilogram per hectare K. And the data that we collected from this was, so we irrigated, and each time we irrigated, we collected the amount of irrigation. The irrigation was derived from a 42-year data from northern Ghana. And each time we irrigated, we collected runoff, we collected drainage, and then we measured the moisture storage in the soil. Each leachate that was collected from each irrigation event, we determined the electrical conductivity, and this electrical conductivity was converted to total dissolved salts using this equation. Some of the parameters, plant growth parameters that we measured were the plant height, chlorophyll, total biomass, grain yield, and then we determined the water use efficiency using the grain mass over the cumulative evapotranspiration. By way of the results, the table here is a snapshot of the result. What I want to talk about is GC here represents gravel content. Whenever you see GN here, it means that we call it non-gravelly soils. Remember, gravelly soils are those with gravel content up to 15%. So when your gravel content is less than 15%, then we classify you as non-gravelly soil. GN here is a granite field where we did the sampling. FAL here is a fallow field. MT here is a millet field where we sampled the soil from. So the first samples shown here are from non-gravelly fields. And what we observe is that all the different water balance components were not significantly different from each other. So a total irrigation of 668 millimeters of rainfall throughout the planting season. And we didn't observe any differences in the different water balance component in this sample. But as the gravel content increased, we begin to observe some differences in the water balance component. Take, for example, we had about 44.28% of the total amount of water that was applied during the irrigation being lost, and the moisture content also decreased significantly, whereas the ET also reduced as the gravel content increased. And this actually became, we saw a further reduction in the moisture content as the gravel content increased. And also about 46% of all the amount of water that was applied was lost as the gravel. So in the case of the disturbed soil sample, so the gravel fractions we packed in the disturbed soil sample range from 0 to 70%. What we observe here is that once gravel content were 40%, anything more than 40% gravel content, the amount of water that was lost significantly increased to about 64% of the total amount of irrigation. And then the moisture content also decreased to less than 0.2 in cases where the gravel content were more than 40%. What we observe here is that because of when gravel content is more than 40%, then macro porosity begins to be the driving force of the movement of the water. Therefore, we had a significant loss of the water in the soils, whereas this resulted in a significant reduction in the moisture content. And also because there is no moisture in the soils, then the evapotranspiration also significantly reduced as the gravel content increased in the soil. To look at the water use efficiency, what we observe is that as gravel in the undisturbed soil samples, as the gravel content increased to 34% in the undisturbed soil columns, then the water use efficiency became high. So the highest water use efficiency was observed in 64% gravel content, whereas in the disturbed soil samples at 40% gravel content, the water use efficiency also became the highest. What we see is that as gravel increases, the amount of moisture in the soil also decreased, but at 40 and 34%, the moisture was sufficient enough to support the growth of pearl millet. And also, if we look at the nutrient loss, which we measured as total dissolved salts in the leachate that was collected, we found out that as gravel content increased to about 64%, then we had more than 50% of all the applied nutrient loss. Similarly, in the disturbed soil samples, at 50% or more gravel content, we had more than 50% of all the nutrient applied was lost. So with the nutrient lost here due to macropores created by the presence of gravels, and that is so, and then also a significant loss in the amount of water that is applied, then you will find out that the water use efficiency becomes very low because all the water is lost through the macropores and also the nutrient. If you look at the grain yield, what we also found is that we found a similar trend of the grain yield being highest at 34% gravel content for the undisturbed monoleaves. And also in the disturbed monoleaves, we also found out that the yield and the total biomass was also highest when gravel content were 40. And we realized that as the gravel content increased in the disturbed soils, what we found is that the yield, the grain yield and the total biomass also increased up to 40%. And then beyond 40%, there was a significant reduction in the yield and the total biomass. As I explained earlier on, when the gravel content is more than 40%, there's a significant loss of the nutrient and also a significant loss of the moisture that is in the soil. Therefore, a significant loss of moisture and nutrient is not suitable for plant growth. This explains why we saw a significant reduction in the yield. to conclude, what this study has shown us is that the presence of gravels in soil affect the water balance component. With increasing gravel content, we realize that the moisture content decreased, but also the water use efficiency and the fertilizer use efficiency increased with increasing gravel content up to 40%, and then began to decrease with further increase and then began to decrease with further increase in gravel content. Disturbing of the soil samples did not significantly change the effect of gravels on water use and fertilizer efficiency, because the presence of gravels already changes the structure of the soil. So disturbance doesn't further destroy the structure of the soil. And soil gravel content beyond 40% created macropores that led to rapid transmission of water and nutrient. We recommend the implementation of water strategies that will be very useful, especially in soils with 40% or more gravel content. Thank you.
Okay, thank you very much. Please switch off the presentation mode.
I've done that. Is it showing now?
I still see it. So as long as
Okay.
okay, thanks a lot. So I can see the group.
Okay.
Well, thanks for the presentation. For me, these results are at least partly astonishing. But before I pose my question, I would like to ask the presenters if they have questions. Doesn't seem to be the case, huh? So,
No, it doesn't seem to be.
Strange.
To be honest, I was busy responding to some question on the app most of the time. Yeah.
Okay, could you see questions in the app? I don't see any.
Yeah, I can see on my phone. I didn't download it.
On your phone? Okay.
Yeah,
I have it. I didn't switch on my phone. Okay.
Oh, okay. Yeah.
So then my question would be for me, Mr. Yanore.
Yeah.
It was astonishing that there is this kind of discrete response chain. Normally, if you increase, for example, this kind of gravel content continuously in 10% steps, I would also expect a rather continuous response curve. In your case, that is not true. With the undisturbed columns and also with the disturbed columns, you get more or less the same response, and you get this discrete step where definitely the response curve changes. And I don't understand why. Do you have any kind of mechanistic understanding why this is the case? I don't understand that.
Okay. The understanding or the explanation I give to this is that, as I showed in the sketch earlier, if you look at water and solute transport in the soil, we can talk about the fact that it could be through the micropores or through the macropores. And, so you will notice that as the gravel contents are lower in the soil in terms of size, in terms of quantity, then they serve as obstacles, or they impede the movement of water. And then the active area for the transport of the water and solute reduces because of the presence of these fragments. Okay. But as you continue to increase the percentage, it gets to a point where the micropores now becomes higher than the macropores. So there's a switch of the effect. So if you look at the results, as you increase the gravel content, the amount of water that was lost was increasing gradually to a point where it became so big. That's the turning point where macroporosity is no longer the driving force, but the driving force is now macroporosity because of the presence of the gravels. So the switch is because gravel content becomes the dominant force, and this brings macropores into the system now.
Yeah, but I don't understand why that should be stable, because afterwards with 50, 60, 70% of gravels, you have the same leaching rates. And
Yes, because the macropores that are created are sufficient enough for the flow of the water and the nutrients. So additional space or additional macro space created doesn't influence the rate at which the solute moves, because the macropores are already sufficient enough to allow the transport of the water and the nutrient to go through.
Ah, okay. Yeah, that's what I could understand. Okay.
Yes.
Fine. Any other questions? No, so I don't see any questions from the crowd. I don't see any questions from the contributors. So I would like to thank you all for your nice presentations during this session. And I'm going to close this session since I can't reach anybody out there else. So that's not my problem. That's just a technical issue. I have learned some things, and I'm that's good.
I think someone was trying to reach to connect, but she couldn't. The third present, I saw her like two times on the top try connecting, but she did not make it through, I think. So
well, we could wait for 1 or 2 minutes, but otherwise it doesn't make sense just to sit here and to wait for something to happen, isn't it?
Yeah, exactly.
Except we could drink together a nice cup of tea.
Yeah.
I only have water to offer. So
okay. All of us have water.
Okay, so let me check for the chat.
Yeah,
Oh, there's no new mentions. Also in the Zoom chat, there's no new mention.
It's strange that we have like almost say 67 person watching.
Yeah. So I would like to ask the crowd if there are any further questions. Please address them to the speakers themselves now after closing the session, because you have the possibility via your app to contact all the participants of the conference directly. Yeah, so you can use your WOVA app, or however you pronounce this app. You can use it in order to get direct access to our presenters, to Mr. Zhu, to Dr. Abdallah, and Mr. Yanore, sorry.
Yeah.
Yeah, and please use this possibility. Otherwise, I would like to close this session. I thank you, the audience, which is a kind of incognito, and I thank you, the presenters. Have a nice day and enjoy the conference in the following days. Bye-bye.
Bye-bye. Thank you. Thank you.